invitation. I should introduce myself because I was very lazy to supply a short CV, and what I did, I put all my my CV point by point, which is 3 pages. Okay, I finished medicine in Sofia, in Bulgaria, and after the 1992 I changed to Switzerland. I did my MD there, and I worked in the clinic. First I was in the surgery, neurosurgery, visceral surgery, and from in the 90s I changed to the anatomy. They made me a really very nice offer, and now I have the privilege to work with so many students to do some research, but to some extent I'm missing the clinical work, and we have a special department of clinical anatomy now in Bern, where I'm the director of the institute, and we have a very, very good contacts with the clinician. We are developing a new technique, operating technique, the new treatment techniques in this department of clinical anatomy, and we are working also very close with the artificial organs. There is a special department in Bern. Today I'm going to talk about a special type of angiogenesis called intussusceptive angiogenesis, and this would explain us why the tumors are growing after treatment. Could I ask you kindly to switch off here the light, because I have a couple of pictures. Okay, thanks a lot. I will start with one slide. I'm showing my students and starting the cardiovascular block, and with the question, why do you need the blood vessels? You could see here immunohistochemical staining of a breast carcinoma, with the staining of CD31 staining the blood vessels here. In a distance about 100–150 micrometer, there is a tumor necrosis. On the consecutive staining, you see that there's a hypoxia-inducible factor 1 alpha. This was introduced today by Hans, and also you could see the necrosis, and this factor is expressed surrounding this necrosis. What's happened? First, you have the hypoxia. The hypoxia induced other factors like vascular endothelial growth factor. This factor is inducing the new blood vessel formation, which is now stained by CD31, or in other words, is inducing angiogenesis. And this on the end is coming back to the normoxia. There is one single reason why we need blood vessel, because the diffusion capacity of the oxygen is a very short distance. It's about 100–150 micrometers. Except the cartilage. Cartilage don't have the blood vessel. There's a completely different situation. And there is the 5 to 10 cell layers. This one is absolutely sure now that it's also logical that if you stop the blood vessel formation, you will stop also the tumor progression. And this was an idea put by Judah Folkman many years ago in a seminal paper in the New Journal of Medicine, and in the 1998 in the New York Times, he postulated that just in a few years we will be able to stop the tumor progression by applying the anti-angiogenic compounds. 15 years later, what is the situation? If you look at that one, it's anti-angiogenic drug in clinical trials. Only in the United States we have 1,800 clinical trials. Worldwide, we have 3,800 clinical trials. No one single substance is working as a single therapy in anti-angiogenic treatment. It's only one is combined with chemotherapy in the colorectal cancer that was proved by FDI. What is the reason? And I will put it a little bit in a provocative way. And as Nenad was in Bern, I showed him that one. And here what we have this is a carcinoma, the breast carcinoma in a mouse model, transgenic mouse model with the oncogene here. And you could see that there's a different vascular phenotype. This picture, they are microvascular cast. You inject the resin in the vessel, digest the tissue, and we have only the blood vessel, and you could observe them by a scanning electron microscopy. But you could see such kind of phenotype, you see such kind of phenotype, coral-like, or this type of phenotype. The funny thing is that all these tumors, they have the same genetical backgrounds. All these blood vessels develops in tumors with the same genetical background. This makes the whole story very difficult. How are we going to model the tumor growth in this tumor? we find out that even in the same tumor, we have two major vascular phenotype. One is looking like that. You see the tumor, the half of the tumor is going like that, the other half is like sheet-like with a small, tiny holes into the vasculature. And there are two types of angiogenesis, and I'm going to give you the short introduction of that one, a historical one. One of the first papers published in the field of sprouting angiogenesis was published in the 1856 by the famous physician Theodor Billroth. And he published using different models like embryonic development, wound healing, inflammation, tumors. And he demonstrated very nicely that the blood vessels, if they are induced, and he didn't know at that moment what is the signal, but now we know that this is the vascular endothelial growth factor. These blood vessels are resemble very close the trees in the spring, and they are sprouting as the trees in the spring. Also, the vessels are sprouting in the surrounding tissue, and he called it this type of angiogenesis, sprouting angiogenesis. What's happened later? These two sprouts they fuse together as here, and on the end this connection is perfused, and the blood could flow through the new segment. This is the concept of sprouting angiogenesis. It's more than 150 years old. But the second one I would like to show you today is a second type of angiogenesis, which we need that one, and this will help us to explain some phenomena. For example, this is the chick embryo. It was mentioned today. There's a model of chick embryo, and this area vasculosa is equivalent of the yolk sac in the mouse or mammalians. And you could see here by 45 hours, you see the very dense capillary meshwork. Only in 8 hours, there is already arteries are here, and this is increased dramatically. I can tell you, you will look at that one very carefully. No one single or very rarely sprouting vessels. How is possible that from that one, only in 8 hours we have a complex now system with arteries, veins, and capillary plexus, and a few hours before there is nothing. And the new concept was introduced in our institute for the former director of the institute, Peter Buri, and he was interested in the lung development. And it's very funny because the mammalian lung increased the vascular exchange surface 24 times after birth, just in a very short time. 35 times the capillary volume. And they looked for, at that moment, they searched for sprouting vessels. They didn't find one single sprouting vessel in that one. And this is a very good institute for morphology. And we did the 1,000 section in transmission electron microscopy, doing us nothing. What they found was such kind of a vascular pattern, or such kind of a pictures. You see, this is the alveoli, lung alveoli looking like that. And in this alveoli, we have a lot of tiny, small holes into the capillary sheets. And here is the hard magnification of that one. And what they postulate at that moment, that most likely, these capillaries, they grow, instead to grow outside, they grow inside in itself, and they create the term intussusception, which means in itself growth. We worked on that in the last 15 years on that topic, and I could represent you the concept of intussusception like as demonstrated here. The sprouting vessel, the endothelial cells, they make sprouts and they go into outside to the surrounding tissue. But by the intussusception, there is like stalagmite and stalactite, they are growing intraluminal. This is abluminal, and this process is intraluminal, the opposite process. And this is going very rapid. The opposite endothelial cells, they are invading the lumen, they are creating a contact, and here is in the schematic picture showing the same process. They are creating a contact in the middle now. This contact is perforated, and the column is inserted in this capillary lumen. What's happened in that one? How this contribute to the growth of the entire plexus? I will show you now. But before to go to that one, because today was introduced the chick choriolampic membrane model, it's looking like that. You could take the embryo, open it in the petri dish, and you have the choriolampic membrane. This is this thin membrane here on the top, and you could see the growing chick embryo lying here in the petri dish. And if you inject now in this classical system the FITC dextran or small bits, you could monitor very nicely the blood flow. And what we did exactly that one, and you could see here at time point 0 there is nothing, the blood is flowing into the small vein, the capillary plexus here, and just focusing through this vessel. But interesting is that area here. After a couple of hours, or very often you are able to see that after minutes, 20, 30 minutes, you see that there is here something arise. Something, a new structure is here in the middle of that vessel. And at high magnification, now you could see that some erythrocytes are coming, crashing here, hanging for a couple of milliseconds, going away. And the nice story being in the Institute of Anatomy and Morphological Institute is that we are able to, you have a very good technicians, and you are able to fix this place and go for cutting, serial cutting, and you could evaluate exactly what's happened at that place. And this is what we did. Actually, we did 100 of serial sectioning here in that area, and you find out the morphological substrate of these structures, transluminary tissue pillar, and now this is a three-dimensional reconstruction of about 200 transmission electron micrographs. You could see here into the lumen that there is a structure going through. In the background here is the erythrocyte, you could see it here, and we could match exactly the time course of creation of such kind of a pillar with the morphological substrate. We know exactly in every one moment what's happened here and what is the morphological substrate. And these, as soon as they appear, the cells invite in this structure and they expand very rapidly. And if you have this process going at the same time at different, simultaneously at different places, the whole capillary plexus expand very rapidly. You observe this type of growth in many organs. You could see here different organs during development. And there is the very old paper published in 1950 showing that one in the embryo here. This is the eye, the brain, extremities. And in the first step, there is a lot of such kind of transluminal tissue pillar, a small hole here. But why, how formation of such kind of pillars could contribute to expansion of the capillary plexus? I could show you a couple of slides now showing the outcome of this process. First of all, if you have such creation of such kind of pillar at the same time, at the many places, they arise, and after they become bigger and large, and the whole capillary plexus simulation is running a little bit slow, but you could get the idea. If you have that one, for example, creation of pillar here, 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 this plexus could expand very rapidly. The nice story is that this type of vascular growth is actually very characteristic by the main characteristic are that the proliferation is a low rate of proliferation, low rate of transformability, a duration of hours. We call this type of vascular growth microvascular growth because it happens in the microvasculature. But this has very important consequences. I will show you now without to go into molecular biological details, and I took out the picture because we could digest them much more easily. In the kidney, this is the normal kidney glomeruli. In our kidney or mammalians, they are looking, all of them, they are looking like that. If you inject the antibodies, as type 1 antibody, destroy completely the capillary plexus in the kidney and the glomeruli. After 3 days, it's looking like that. We have some capillary segments, the rest is gone. What's happened? How this is now reorganized? You have two possibilities. One hand is the sprouting, other hand is intussusception. What you are, most likely they are sprouting vessel also, but in the background you see a lot of small, tiny holes here. By day 5, they become more and more, and by day 9, you see here many tiny holes by high magnification. They are inserted into these capillary segments. What's happening with these pillars with these tiny holes? What they are doing, they reshape, they elongate, and they start to fuse together. As here, you could see here, small pillars. This is, I did this drawing to make whole process understandable. Now all these pillars, they reshape and they fuse together, and now they are cutting like a zipper, like opening a zipper, you are cutting from this capillary plexus new capillary segments. And this is the way how not only the volume of the capillary is restored, but also the capillary part, the organ-specific angiogenic pattern is restored. The process is also very important for the formation of arteries and veins. The same picture, how these arteries and veins arise from this capillary plexus. Nobody knows. I can show you now a picture. For example, this is the eye, the vasculature of the eye. You can see here the iris, the pupilla, the vorticose vein. How this complex capillary system arises. And this arises from that one. And by day 5 of development, the vasculature, the whole is looking like that. One two-dimensional capillary layer. Nothing, no supplying vessel, only 2 trunks here. But by some fantasy, we'll appreciate that here some of the pillars, they are placed in rows like that, or maybe here. This is only idea at that time point. A few days later, at day 10, you could see that all these small transluminary tissue pillars, they reshape and they become slits, and they actually now they are now cutting the former small supplying vessels. If you look from outside, the capillary plexus from outside, you will see that one. Now they are the fusing pillars here. They are fusing and they are segregating supplying vessels, small arteries and veins. And on the end, if you don't know how this happens, you will be never able, if you don't have the dynamic in this process,